Good afternoon. Welcome to this edition of the NITheCS Colloquium. I see some people that I don't really know. So my name is Francesco Petruccione. I'm the director of NITheCS. Yeah? And thank you very much for finding this venue, because usually we do the colloquium in the NITheCS on neutral grounds. Yeah. So today it's a pleasure to introduce Sandra MacFadyen. Sandra is known probably to all of you. Yeah, that's probably the reason why you are here. Yeah, she's, I think, formerly a postdoc in the mathematics department here in Stellenbosch, and she's a NITheCS associate, and she's part of one of our NITheCS research programs, where she leads one of the work packages. And usually she spends her time where we would like to be, in the Kruger National Park, but this week she's here. Yeah. So, Sandra, thank you very much for accepting the invitation to give a talk, and we're really looking forward to your presentation. Thank you very much. Thank you.
Great. Thank you, Francesca. And thanks to everyone for being here today for my talk that I'll be discussing turning data into decisions by bridging mathematics and ecology with policy. I'd like to start off by saying that the work I'm presenting today is the joint vision of a number of individuals, not just my own, especially those from the rest of the Mathematical Biosciences group, BioMath, as well as NITheCS and the BCube team, which is an EU-funded grant. And just to give you a brief outline of my talk, it's going to be split into 5 separate sections, beginning with just an introduction and context. And there I will give you some brief background about myself and dive directly into how we go about turning these data into decisions, as my title suggests. I'll then go into a section on collaborative science. I think that's really important. All of us will agree that there's transdisciplinary and multidisciplinary. All of those names we've given it is a hot topic at the moment. And I'll talk briefly about the different teams that we've been working with. That is the BioMath group, the NITheCS group, and the BCube group. And I'll share with you our joint vision for harmonized biodiversity data, metrics, workflows, and all of that. Essentially, the underlying principle of all of these outputs is a unified framework. And we've found that in something we call biodiversity data cubes. So I'll discuss that in detail and jump across to what FAIR means in terms of data sets as well as workflows. I'll then talk briefly about some of the tools that have come out of these different programs and projects. And I'll highlight some biodiversity indicators, which makes those metrics accessible across different scales, and then the different workflows and tools that are accessible to students and those who want to learn about it. If we've got time, I will dive a little bit deeper into 2 of our main products, being our dissimilarity analyses and the invasibility analyses. I'll end off just with some highlights for capacity building, some of the stuff we've done, some of the stuff we plan to do, and then talk about some future directions. So first, a brief introduction and some personal background. I started early, interested in nature. Sorry, that's Matt. So my dad worked for Natal Parks, or the KwaZulu-Natal Parks Board, and I grew up in the bush. And during that time, I developed a connection with nature, as you can imagine. So after school, I studied nature conservation, and I was lucky enough to get one of the research positions in the Kruger National Park in Skukuza Camp. And that was an amazing opportunity because as a research assistant, research student, you get to go out with all the different researchers, botanists, ecologists, animal behavioralists, and basically everybody. And I spent the next 15 years, essentially, so I'm giving away my age, working and living in the Kruger National Park as a geospatial analyst. And during that time, I was able to further my postgraduate degrees in nature conservation, and I went on to do an MSc in geographic information science. I soon realized when I started my PhD that working and PhD at the same time is a bit tricky. So I decided to resign from my position to pursue my PhD. I had a joint PhD with Stellenbosch University and the Vrije Universiteit in Amsterdam. And it looks like Randolph and we, the hobbits in that group, the Dutch are tall and so is Kang. But my thesis was on ecosystem interactions in a heterogeneous landscape. And through that, I was able to get a better understanding of modeling and spatial temporal dynamics. And so, as Francesca mentioned today, I'm a postdoc with the Mathematical Biosciences Lab at Stellenbosch University at the mathematics department. And my research focuses on macro-scale ecosystem dynamics. So it's more at a landscape level, looking at the patterns and processes that we can find. I'm also interested in the interactions between environmental drivers and responders, and how do these dynamics shape and sustain the landscape patterns that we see. And then ultimately, I'm interested in developing effective and efficient and economical tools for tracking macro-scale biodiversity patterns and trends. So conservation agencies, we never have money, and there's a lot of really cool gadgets and things out there that we could use, but we need to find something that's more practical, that can cover large areas, protected areas, and something that's economical. So with an interest in such a wide topic, like spatial temporal dynamics, my research focus has been quite varied, from landscape patterns, as I mentioned, through to animal movements, rainfall, fire, different remote sensing products. And this has enabled me to work with some really amazing scientists and had some really amazing PhD students working with us. And because this is such a wide focus area, it's also allowed me to work on projects related to invasive species, community turnover dynamics, even some genetics, as well as a more holistic ecosystem assessment. More recently, I've been thinking about how we're essentially drowning in data, thirsty for information, yet starved for understanding. So in this information age, I feel a multidisciplinary approach is essential to turning data into decisions. Natural sciences in particular needs to connect with more fluidly with information or data science. So in this paper, what we actually did is we used this Nautilus shell to depict the transformation of data observations. Let's see if this pointer works. For the people online, they can see it. In point 1 there, the data comes in. That's the raw data. There needs to be some sort of system in place to design the data management plan so that we can standardize all the data. If you can imagine multiple researchers doing different studies, they collect it in different ways, different structures. And for you to be able to use that data, it all needs to be in one place in a unified format so that you can start making decisions and finding answers from multiple data sources. Data centralization, this is essential in terms of accessing the data, so not just for a research perspective, but also for policymakers, and that data services are then needed to be able to serve those data and for people to inspect. And once that's done, once the data is essentially been formatted and served, it is out of the system in a way. People can then use that data to make their own interpretations, to feed it back, hopefully, into policy. But that is, I think, a big gap. And we're trying to fill that gap by creating workflows that allows users to bring their own data and follow a similar process to look for answers at different scales. So if we have a data set that covers the globe and we run it to look at community turnover, and you have a very small data set in a local area that you want to look at turnover, you can bring it and run it through the same process and it will give you a result. Having said that, it's really important to keep the ecological context so it doesn't what do they call it, rubbish in, rubbish out. If you don't have the data collected correctly and you don't understand the outputs, you're obviously going to have a problem. And yes, that process actually, through the help of NITheCS, we were able to create this special issue looking at biodiversity informatics and the importance of it. And it helped showcase a few of the projects where we could see how important it is for us to start standardizing these data and centralizing it so that we can start making sense of it. So in other words, our understanding of ecological systems, for it to be more holistic, we need to combine these different disciplines. And in the past, you've had ecologists or research ecologists sitting in protected areas studying different phenomena. Maths has been over there looking at chaotic systems and developing formula for sometimes abstract ideas, but it's in the middle that will happen. And by bringing those two together, we've been able to make some advances in the field, but we've only touched the surface. And hopefully today, my talk will help everybody start thinking about those, because what's more chaotic than nature? So if you're studying chaos theory, it's still relevant. And I added this in quickly, that this is a new part of it, that the data science is a really large part that we often skip. And we don't need to be experts in all the fields, we just need to work with good people. And I think that's the key here, is to bring the people in to work together. And without that, without being able to serve these data and put it in a digestible format, it's never going to feed back into policy. And I think that's something for conservation, especially if that doesn't happen, we're in danger of losing a lot of our protected landscapes. So just to talk a little bit about some collaborative science and the different groups that I'm working in and how they fit together. We've got our BioMath group. As I said, we're a dynamic group of researchers that are led by Prof. Cancri. And we work at this interface between mathematics and biology. Our interests are in developing models and theories for explaining emerging patterns in ecology and the ecological processes that are so highly complex and adaptive. We really rely on the simplicity of the mathematical language to build the models and theoretical frameworks. And we're all friends. I think that's a key to real collaborative outputs, is that you have a trust built that we can work together and we can produce things. And just to prove it, we at the same time try to spend some time balancing the screen-based work and our modeling and mathematics with some field engagement. So we do some periodic field trips. We actually came back from one last week. And yeah, there's even applied maths playing with chameleons and geneticists looking at dung. Vernon, you're also there jumping off a cliff. So yes, I think that's really important to keep grounded with nature, especially in an age with remote sensing. It's so easy to just go online, download a data set and start working with it. But the reality is that those data sets are often built at global scales and they've been calibrated to those scales. So if you're working in a very small area, the chances are that those models are not correct. So you need to try and validate those with ground data. So then our NITheCS research program, as I mentioned, we were able to produce the special issue through Frontiers, the Frontiers Journal. And we've switched now from our original program into the E5 program, and the work package 1 is Ecological Complexity and Biodiversity, and I'm helping lead that. And our aims there are to develop our understanding of these biodiversity and the complexities within it, and exploring and unifying measures that can help us speak the language of biodiversity and look at how it's changing over time. We've also had, through NITheCS, we've been able to do a number of workshops and seminars, which has been really useful in helping explore some of these complex ideas. We've done a few micro schools, looking at some more technical approaches. And then we've got our own mini school that we do every year, where we look at practical ways of creating biodiversity data cubes and how do you go from there, and different techniques that you can use. Then we're a partner in the Horizon Europe research project, Biodiversity Building Blocks for Policy, commonly known as B3 or B cubed. And there our mission is to help transform biodiversity monitoring into an agile, policy relevant system using data cubes, but also open workflows and real-time indicators. So that's a massive program that I'll unpack a bit further, showing you some of the deliverables. Yes, so that program itself, we're actually 9 different countries with 13 different institutes. Stellenbosch University is one of those in South Africa, along with SAMBI. And I think through this group particularly, you can see the benefit of collaborative science and having a joint vision in this scenario towards harmonized biodiversity metrics and these open source workflows. So together, our joint vision is rooted in the premise that biodiversity is the foundation of a healthy ecosystem, and that we're in the midst of a global crisis when it comes to biodiversity loss, with threats like habitat loss, climate change. We need to make monitoring biodiversity relevant for various scales. The example I gave to you earlier about a global versus a local scale, we need to have measures that can be used across those scales. And yes, to do that, we need a scalable approach that measures this change. And it's not only a spatial scale, but a temporal scale. So through the BCube program, we've highlighted the different challenges and opportunities linked to this. Once again, looking at rapid, reliable, and repeatable biodiversity monitoring data, which decision makers can use for evaluating policy. And through that, we're able to help move this challenge forward. We have a number of solutions and activities that we highlight for you briefly. Once again, a large component of that is policy alignment, which means stakeholder engagement. We have to get everybody involved into the same room to discuss these issues, and making sure that it's even evidence-based. So we have monitoring strategies that are local as well as global. So I'm going to go through some of these in more detail, but essentially there's the automated workflow section. We've got some cloud computing and the capacity building that's important, and we have case studies which I think are really nice to be able to see how one uses these outputs in a meaningful way. The different work packages within the BQ program, there's a number of them. I just want to highlight these 3. I'm mostly involved with the modeling current and future scenarios under the work package 4. Mukta and a few other members are looking at creating indicators of biodiversity change, including quality measures, and then the implementing case studies, which we work closely with SAMBI on that. So, why do we need all these groups to work together, and why do we need all these different frameworks? Well, biodiversity, along with ecosystems, is complex. And just to give you a quick overview of biodiversity, I'm sure most of you are aware of it in the room, but biodiversity is essentially all the variety of life on Earth. But what we often forget is that it's also all the ways these living organisms can interact with each other, but also with the environment. So if we've already established that it's species, there are different scales to this. So not only species diversity and this variety of life, but also at a different scale, you have genetic diversity, which is the variation of genes within species, which allows for adaptability and resilience. Then there's ecosystem diversity, which is a variety of ecosystems, things like forests, oceans, grasslands, those that provide different habitats and services. But even further, there are different components to this in itself. So we spoke about species. You can have a composition, which is identity and variety of species, but it can start with genes, populations, ecosystems, land types. So everything is nested within these different components. So you can already see the different scales that we're looking at in terms of understanding biodiversity and how intricate it is. Then you got structural, which is the physical organization or the pattern. So that again goes through to genetic structure, population structure, ecosystem or habitat structure, and landscape patterns. Another step which comes to play when we look at the interactions often is the functional diversity. Sorry, I thought that was me. Ecological processes and roles that species and genes and ecosystems play. So that's genetic processes, population dynamics, intraspecific interactions, and landscape processes. So I think I've shown you that biodiversity is complicated or complex, and it has multiple components, scales, regions, and taxa. So as we build it up in this figure we see here, it's very simplistic, but in actual fact, it's a lot more complicated than that. And to give you an example and to illustrate this, if we look at the landscape, one could look at the species composition, looking at the variety of vegetation that we have, or we can also look at the structural composition, how many large trees, how many shrubs, grass, how many bare grounds, all of these providing habitats for different species. And so on and so on. You can look at a variety of different components within the landscape. But what's hidden is always these interactions that you're not always seeing, and that's where the complexity comes in. And with those interactions, they're all linked. So I often like to think of it like a spider, where you pull one part of the web, you're pulling another part of the web. And this is doubly important when it comes to looking at your results, because you can produce a model that works perfectly for the Kruger National Park, but it's not necessarily going to work for Addo National Park or Provincial Park. No, it's a national park. So yes, you need to make sure that your methods are transferable. So how do we do this? How do we measure and monitor this complexity? And as I said, we need a unified structure. And DataCubes has offered this structure, or at least a view of using this. And just to give a rundown on what a DataCube is, this might link us nicely together from an ecological as well as a maths perspective. They're multi-dimensional arrays of data. So they're often used for spatial temporal analysis. So I've used this often in a raster setting. So they have at least 3 dimensions, either firstly time, space, and then some sort of variable, whether it's rainfall, animal movement, or something like that. So let's give an example. We have a variable that's rainfall. It changes every year, and it also changes where it rains. So the latitude and longitude, and the bigger region is Kruger, for example. So often you would get papers or research like this where it tells you the temporal trend of rainfall. It will tell you this is what's happening to the rainfall, and you can look at the El Nino and other effects. You can also look at spatially. You can say, all right, this is where the rainfall generally falls, so this area is wetter or drier than the other. But it's changing all the time, and that's where we often are limited in our understanding of it. And not only that, there's also the interactions that I was talking about. So if it rains more in one area, there'll be more biomass, which then can affect grazers and browsers, for example. And not to mention added disturbance factors like fire, for example. So as you can imagine, it adds up quickly. So just an example of a matrix, you can have the rows and your columns, which we can say their latitude and longitude. This is one matrix for a year, say it's rainfall. You can add on a few other years, and we can do that for fire, temperature, animals in the landscape, NDVI, which is essentially vegetation, biomass, rainfall, and even human disturbances. And they're all linked in this common format. But the issue is you can't just add them all up. The sum are not equal to the parts. And this pretty picture of what it looks like in the environment, we have to keep remembering these interactions that are happening. And there's not just one interaction, there's multiple interactions. And those interactions influence elements as well as other interactions themselves. So how do we account for these interactions between different scales like genes, species, and communities, as well as their environments? And how do we add these up across time and space? And that's where mathematical modeling comes in. And this way we try to simplify ecological complexity by quantifying the processes. So I'm an ecologist by training, so that's my eyeballs when I first started at maths. But it's not so scary if you work with good people and you trust and develop methodologies with those people. And it is a new avenue that we can explore, and we should. Just some basic examples of what we're working on in our group. The emergent ecological patterns. So those patterns revealed through modeling, just a simple example of looking at landscape structure, for example, delineating these different communities. But then you've also got these adaptive ecological networks as animals move through the landscape, and they're influencing it as they do that. And then things like biodiversity turnover across scales. So mapping the spatial and temporal change, and not necessarily just turnover, but change in general when it comes to diversity and biodiversity. So to track biodiversity and environmental changes across these scales, we do need data. And the quality and the analysis of the results rely on this well-structured, standardized biodiversity data. And to effectively use it, we need to employ more data science techniques. And this FAIR approach, which is findable, accessible, interoperable, and reusable, comes from those fields. And if we don't apply those, we won't have a consistent data set or unified data set that we can explore. So through the project B Cubed, as well as our BioMath group, we have some solutions that we've been trying to explore in terms of biodiversity at different scales, two of which are the biodiversity data cubes and the biodiversity indicators. So they're all based on a unified structure of a data cube, but we're essentially creating these pre-aggregated and modeled biodiversity data or data cubes that you can explore yourself, as well as developing standardized biodiversity indicators of change that you can also explore. So the different cubes that we have available are things like the species occurrence cubes. So this is essentially a tab separated CSV files, a matrix that has species occurrence measures in it. You can have count data that come directly from GBIF, or you can start looking at different indices like richness and evenness. So all of these are accessible. I'm going to show you in a minute how you can access them and explore them. Then you have the biodiversity indicators. So these are cubes that are used to generate these indicators that translate this complex data into clear measures of biodiversity status and trends. So a very simple example is the richness species. That is an indicator of health. And we've tried to employ some case studies, but we're developing a lot more using these data so that we can show that it does work, or if it doesn't work, it doesn't work. And these are just a few of them that we've got set up at the moment. They're almost done, but we're going to be adding more to the plate. And again, as I said, policy alignment, it has to happen. And it's difficult to have it happen because you need to have a stakeholder engagement. You can't have a group of scientists sitting in a room saying that A, B, C, D, E, and now make it happen. We can't make it happen without policy. So we've put a lot of effort into trying to do that, linking stakeholders together and showcasing some of the results. Things like the Sustainable Development Goals, for example, and trying to input data directly into those measures. So this is the website. This is the GitHub site for BCube, this biodiversity building blocks. It looks like that. If you go in there, you'll see a number of different repositories that you can explore. And just to highlight some of them, outside of the BCube environment, because we've got a number of stakeholders, we work with them closely to have our deliverables worked into those. So things like the RGBIF, if any of you have used GBIF data, so that's spatial occurrence data for species, you can use the GBIF package to bring your data in from GBIF into R. You've got the EVCube, which is an R package that lets you access those essential biodiversity variables from GBON. So that's important for tracking change. And then the Triage, which is the R package tool designed to support tracking invasive species. Then you have our B3 verse, which is the B3 universe that we put all of our products into. So if you go into this website, which I'll show you in a minute, you can access all the documentation on how we develop these different cubes, as well as access the readily made data cubes. Then you've got different systems that would look at the data itself. So G-Cube, where you can simulate your own data cubes and start playing with how they work, as well as DubiCube, which is another package that helps you understand the uncertainty around the data that you've collected, especially if you're accessing it through GBIF. And then we have our different indicators, things like the PD indicator, which is your repository for the phylogenetic diversity indicator, and there's a workflow for that so that you can input your own data if you have it. And then the IMP indicator, which was developed by one of our PhD students. So that's looking at computing impact indicators of invasive species. And that's directly working with SAMBI as well. And then the two model data cubes, which I've been working on mostly, is the DisMapR, which is a R package for compositional dissimilarity and biodiversity turnover analysis, as well as the InvasiMapR, which is a workflow to visualize trait dispersion and assess invasibility. And then we're still exploring other ideas for different indicators and different workflows. So if any of you have any ideas, please come and chat to me. We want this to be a system that can help from a student level up to a manager, up to a policymaker. So just to give a quick rundown of these different tools. So I know not everyone knows what GBIF is, and not everyone is working with species records. But as I said, this is an R package that fits within our program that pulls data from the GBIF. which is a repository for all biodiversity data species occurrences. And in there, we've got, because we partnered with them, we've actually set up some workflows on how to introduce these biodiversity cubes to the GBIF community, and so that you can develop them yourselves through their interface. Then this EBV cube, any of you that have been working with Daikini targets, you'll know about the essential biodiversity variables. So these are global indicators of ecosystem health and biodiversity. So we partnered with them as well, so that you can use that data portal to access data. And then in the invasive species tracking, so this is a separate ARC package that's part of our collaborations, that's providing functionality for tracking invasive species. And then as I said, the B3 verse. So as you go through our GitHub site, B3 verse, you'll see this website that brings up all of the different products that we've essentially created. And the documentation section, I think, is really useful. If you go through there, you can learn on how to work with biodiversity data cubes, and you can look at all of the components of it. So on the left-hand side there, you'll see all the guides about the different cubes, as well as all the different software packages that are in there, as well as some tutorials on how to develop different analyses. So those tutorials are always growing. Keep an eye on that. And if you want to have something more relevant to you, we must chat about it. And we've got the B3 data. So as we've been working, we're also putting data resources for this B3 verse, so that if you don't have your own data, but you want to be able to see how these processes work, you can do that. You can just access it through the B3 data package, or you can use the RGBIF for other versions. There, we've also put a lot of the scripts. So if you want to go and use your own data and develop these different data cubes, you can do so. And all of this is also getting uploaded to Zenodo. So all these data are available through there. All right, so then we've also got, oh, it jumped up. Yeah, sorry, so G cubed, this is the simulation framework. So if you need to simulate any data, you can use these. And DubiCube, which I mentioned earlier, is a package to explore and visualize uncertainty around it. And this is the BCube General Biodiversity Indicator. So this allows you to analyze biodiversity trends and spatial patterns directly from GBIF data. And the phylogenetic diversity indicator, also a lot of information there. As you can see, it's all built around a common workflow that one can apply. The IMP indicator, which is your impacts for aliens, alien species. This is one of our PhD students that's worked on this. So with Stanby. And then the model data cubes, things like this dissimilarity framework for compositional dissimilarity and biodiversity turnover analysis. It's a package, but it's also a workflow so that you can work through it with your own data or with simulated data. And InvasiMap, which is novel framework to visualize trait dispersion and assess species invasiveness and site invasiveness. So I've thrown a lot of sites at you, but essentially that's the GitHub site where you can go and play around with a lot of the stuff. And please do, because it would be great to know if you can break it. If you can break it, we need to fix it. So have a look and play with those workflows and those datas. Here's the QR code if you want to look at the cubes themselves and how do you download these species occurrence cubes. And I don't have much time, so I'm not going to go through this compositional dissimilarity and invasibility packages in more detail. The website is there, and it will take you through the process, and my details are on there should you contact me. Right, so that's invasibility. So last thing I want to talk about is just the training and capacity building. This is a very important point, especially because we've designed them in a way that students can follow. So we have also different GitHub sites through NITheCS as well as our BioMath group, where we've slowly building a whole selection of tutorials and workflows. And so if there's something you're interested in, or you've worked on a tutorial yourself and you think it's relevant for biodiversity analyses, chat to us. And through B-Cubed, we're also fortunate because we have a agreement with Pensoft. So we've got some really nice website designers out there that are helping us set up these different guides and tutorials. So on that website, you'll be able to see different ecological modeling workflows that are not necessarily related to data cubes, but are still relevant. And we've been able to do different trainings through NITheCS, our mini school, as well as some other workshops through B-Cubed. So the main point is, hopefully I've highlighted to you how collaborative science can really work. But for that to work and continue, we need to have open data and open code for reproducible ethos. And these are the groups that I've been working with, and we've developed some really cool tools, I think, but you in the room are also potential collaborators. So I'd really be happy to chat to you. And for the future, we're looking at scaling up. We're going to look at some machine learning and AI in collaboration with applied maths. We're also developing some dashboards that are easy for policymakers to click in, click out, and export. And yeah, we're coming up with new data cubes all the time and new workflow ideas. And yeah, before I end, I just wanted to dedicate this to the memory of Nick Zambatis. He recently passed away last week, and I just leave this slide up for you to read. All right, then I can take some questions, and thank you.
A very informative talk. I'm sure we have lots of questions. It's nice to see that the signal filled up during the talk. Any questions?
We need to.
Sorry?
We must move this around if someone.
Some questions. Here we go. Sorry, so that the people online can also hear.
When did you guys start working with them?
It's been going now for roughly 2 years. So we have a 3-year program. It ends in August next year. But the BioMath program or BioMath group with Ken Queen will continue. So I'll still be now. I'm very interested to explore more ideas.
That's in good order. Yeah.
I'm now interested to know if either somebody from mathematics or someone from biology wants to use this framework. How steep is the learning curve? Is it something that you take 3 years to get into, or is it user-friendly? Are you supposed to be able to use it within months? Just an idea of where it is pitched.
And then you go up. And then you go through the processes of the entire framework, beginning with biodiversity data cubes and then ending with analyses, workflows. I think it's quite intuitive, or we hope it is. But it would be really great if you could play with it, and if it's not intuitive, let us know so that we can adapt it, because we want it to be something that people can use at any level.
Steps.
Thanks, Francesco. Thanks for your presentation. So are you doing anything in reforestation? I'm just curious because, yeah.
So that is actually a very good example of where remote sensing can fail us. So a lot of these deforestation products that you get and reforestation plans, at one stage they suggested that we replant Kruger with pine trees because we need to increase forests. So we're not doing something specifically with that. We've got some groups with agri-science that are looking at forestry specifically, but Kruger has a very small amount of forest, and the riparian forest is something completely different from what's down here in the Cape, for example. So I think that we need to be cognizant of that as well, and know the environment that you're working in. So I hope that answers the question. Sort of went off on a tangent.
Yeah.
In your presentation, you did mention that you are currently working with some schools. So I would like to know, what criteria does a school that you work with, or what sort of data that you look into so that you can work with that school?
So I myself haven't worked much with the lower level schools, so your primary school level. We have done some outreach in the neighboring communities outside the park, so I have been involved with that. That's less to do with understanding data cubes and more to do with understanding biodiversity and geography. So what we do then, we go into those more rural schools, and we try to bring with maps and things that we leave behind that you can use as educational tools. And then we have little exercises with the kids to figure out how does biodiversity fit together with all the animals that they understand in their area of interest, for example. And in that way, we hope that getting students like that interested in biodiversity at an early age, they want to continue their studies and protect it. So in that sense, that's the experience I've had with it.
Sandra, there's a question online by Douglas. It says, is the cube concept simply a way of showing data, or more dimensions, or something more?
The data cubes. So what I presented today is just data cubes as a unified structure. And because we're linking all of these parameters together, it's a way for us to do this interlink. So in our application, it is, but obviously there are different ways in which you can apply.
Sandra, thank you very much. My name is Carlos Ibanez. I am a fellow at NITheCS currently. I have more of a layman question. You mentioned invasive species, and maybe if you can give us some examples of invasive species in the Kruger, for example, that have recently have to be dealt with. Which are they, where they come from, and what are people doing about them?
So yes, invasive species, you've probably heard interchangeably invasive species or alien species. So there's a difference between an alien species versus an invasive species. An alien species is one that's been introduced to an area where it doesn't occur, and that has different scales, whether it be endemically or nationally. So if you have a plant that comes from Mexico that comes into the park, it can potentially do harm to indigenous species. It only becomes invasive if it does that, if it starts to spread uncontrollably and homogenize the landscape, essentially. So in Kruger itself, often when we map those distributions, it's linked to pathways through the park. So things like rivers, where they have to run through the boundary and they pick up seeds from outside people's gardens, industry, and it runs through the park, gets deposited, and then they start growing. So a lot of our monitoring is focused on those type of areas. It's also roads. Tourists bring in seeds that they don't know of, or they bring in a spider. It hasn't happened in Kruger, but it is possible. The same thing with these shock water beetles. They came in through ports on wood. And so we have a list of specifically plant species that we're prioritizing. As you can imagine, in Kruger, it's 2 million hectares. So to find these plants is not always easy. And that's why understanding the pathways of entry of rivers is important to focus efforts. And yeah, the animal and yeah, there's also a snail, wild cats and things like that. But those aren't really my field.
Are there attempts to remove them or just monitor them?
So a lot of our efforts is in biocontrol. So then you have a species that's been tested for multiple years. It's supposed to be tested for multiple years. And then it's released onto those plants and it allows them to be controlled naturally. And so spraying them, you have to choose either biocontrol or spraying, because if you want to spray the biocontrol's food source, the invasive species, you're going to kill it. So it maintains it at lower levels, but essentially once it gets to an invasive level, it's very, very hard or impossible to eradicate. It's more sustaining the levels to a natural state in a way, not so that it becomes homogenization.
Thank you for the talk. I saw some extreme mapping skills that you had there. I don't know what do you use when you're mapping, but I saw that it is very detailed. Do you also, when Kruger, do you use ArcGIS too, or and not only just that, I saw also there was a way that you beautifully displayed it. Also, there was like an interconnectedness of, like, you said it very well, that certain, it's so detailed. Now, my question was also behind the ArcGIS is, who does the outsourcing for your data? Do you have like a third company that does that for you, or is it in-house, everything with the data collection and the shape files and all that?
Well, first part of your question, we do use ArcGIS and the free packages QGIS. I trained on ArcGIS and the Esri products, but I've switched to R and Python now, so I'm trying to keep everything open source as far as possible. The data collection, it's not just me or my team. That's where the collaboration becomes key. So people in the protected areas, they've been collecting data for years and they use it often for their local management, but it's not always fed into a bigger system. So it's important to collaborate with people like that to bring these data together. And then the techniques that one can use to do that, things like data cubes and these workflows through R. So I can't stress enough the importance of collaborating, because it's not just one person that can do all this. It's a lot of people from different disciplines.
With the data that you collect, is it also open source for what you gather, or is it?
So as you can imagine, things like rhino populations, we wouldn't make that available to everyone to use. So then you would have to apply for access to that data. If you're going to do research in South African national parks, there's a project registration process that you have to go through, that you detail what project you want to work on, and from there you can access those data. All the data that we're working with through these programs are open source, so you can access them.
There is a question online. So Douglas, you're welcome to unmute and ask your question.
Thank you. I'm just trying to grasp the essence of what you're offering. Am I correct that it is basically through the cubes, a convenient, efficient, well-developed method of presenting data graphically? But from there, it's purely from that. The question of how you move from there is a question of just facilitating hypothesis development and testing. There's nothing specific about necessarily hypotheses, the hypotheses and the testing themselves. This is essentially just a question of appropriate presentation of data. Thanks.
Thank you. Yes, so it's both. So we're in the final stages of the project, so we're slowly uploading all of our outputs. The first of which is that you're seeing a lot of the baseline frameworks and creating these data cubes, but essentially it will grow from there and have these different hypotheses that you speak of. And those hypotheses need to be driven by stakeholders. So there are a number that we've identified and we're going to run them, but we need input from people on the ground to see what's important so that we can develop those methodologies to answer those questions or address those hypotheses that you've discussed. That answer your question?
Yes, as far as you've taken it. If I could just quickly add one more, because I think the essence or the real difficulties come when in the process of using what you've got here to take forward, when you talked in your title about informing decision makers, how you move from this efficient presentation to policy decisions. I'll give one example, and you talked about indices of ecosystem health. But it would seem to me that when you come to ecosystem health, the whole debate revolves around how you define that ecosystem health. You'll get very different answers depending on how you define it. It's not necessarily that you're going to easily get robust definitions of such a concept. So you're facilitating something, but am I correct? You're not really offering, without further work, an advance in how to actually get robust measures. Thanks.
As I mentioned, yes, we're building on it. So at this stage, whether it's robust at the scale that we've done that study. So you're 100% correct that whatever we create here is applicable to that scenario. And that's why we want the workflows to be in place, so that it details exactly under which conditions these workflows can be applied, and that we need to remain cognizant of the fact that anything that is done in a specific area is not necessarily going to work for another area or another situation. And as you've correctly said, that we need to be cognizant of the scale that we're interested in. Are we interested in the entirety of it, genes, species, landscapes, and how do we bring those together? And that's in the development of our indicators. But an indicator is a model, and I don't know who said it, but all models are wrong. Some are just less wrong. So the only thing we can do is really detail exactly what's being done and try to build from there. And if there are specific questions that you have and different ways that you could envision combining this, it would be great to chat. But we are in the process of also exploring how do we combine these different levels to track change.
Okay, thank you.
Thanks. Okay, if there are no more questions, then Sandra, thank you so much for our great talk.
No,
This issue of, and Sophie is here. Yeah, our famous editor of this article.